Olá, bom dia a todos. Obrigada pela audiência. So I will shift to English in honor of our inviter today. That's Professor Amin. I met him kind of 5 years ago in a congress in Thailand. And so we are honored today to have him here visiting São Paulo for the second time. Dr. Amin received his degree in electrical and electronic engineering from Bangladesh University, and in electrical electronic system engineering from the University Kebasgan, Malaysia. I'm sorry for the pronunciation. And he's actually, and he has PhD degree from Nara Institute of Science and Technology, NICE, where he's still working today. So we are very happy to have you here. So please, the stage is yours. Thank you.
Thank you, Professor Hayashi, for nice introduction. So I'm going to talk on a long title. As you can see, that it is novel methods and tools for clustering of simple and bipartite graphs. This can be simple and bipartite graphs, and I will talk about the applications in ecology and some computational biomedical research. So all these topics might not be so much interesting to students of pharmacology because this is mostly bioinformatics related works and maybe computer science oriented works. So I will focus more on the topics that are more related to pharmacology. So to do that, first I will talk on a database, knapsack database. This database we created, or we created it in our lab, and this is a natural product database, so it might be interesting to the students of pharmacology. And the concept of knapsack database is very simple. We try to accumulate information about species and metabolite relations. That means which species produce what type of metabolites. So metabolites, sometimes we call them natural products also. Currently in our database, we have 160,000 pairs of species and metabolites, covering more than 51,000 metabolites and roughly around 23,000 species. And we started to build this database in 2004 with the target to finish by 2010. And as you can see, initially our pace of accumulation of the data was not so high, as it is shown by this red line. And at some point, we took the help of the computers and tried to apply automatic duplicate checker in our data. And then we have been able to finish it several months before the targeted time. And all the data we accumulated here from published literatures. So we have to explore a lot of papers related to natural product and their origin and their activities and so on. According to this reference of 2003, the predicted number of natural products in the plant kingdom is around 200,000. And according to this reference of 2000, non-natural products are roughly 50,000. So currently in our database, we have more than 51,000 metabolites. So we can claim probably that in our database, we have the information of all the known natural products. And I think as a student of pharmacology, you know about this. These are according to this reference. We human, we need around 2,500 metabolites to survive for healthy survival. But the plants need more metabolites because they cannot move from one place to another. So they need more metabolites to survive, to cope in different adversarial environments. And the bacteria usually need around 800 to 1,700 metabolites. So this information we accumulated, we did a lot of hard work to accumulate such informations. And we are, this database is freely available to anyone. So you just search the internet with this word knapsack database, and you can have access to our database. This data can be utilized for basic study in metabolomics or to understand the pathways of natural products, evolution of the pathways of natural products in different species, and also gene to metabolite relations. So we are applying, and you can also apply this database for different types of applied works like food science, health creation, herbal medicine, and drug development by herb, or so on and so on, these type of works. This is if you go to the internet with the knapsack family database, if you search, you will get to this window, and here is the core system. By clicking here, you can access to the species metabolite relational database, and also in our lab we have developed some graph clustering algorithms about which I will discuss next. But here I want to say that by clicking this button, you can have our tools of graph clustering. And in this database, actually, we have accumulated other information like healthy diet and tasty diets like food and health, and crude drug means that Indonesian traditional medicine, Jamu, and Japanese traditional medicine. Also, we are now working with Chinese traditional medicine and subcontinental traditional medicines like Unani and Ayurvedic medicines. We will add those in this section soon. And currently, I'm also working on the biological and medicinal activities of the metabolites. And we also have a metabolite ecology database where we have information about volatile organic compounds. So volatile organic compounds are also very important for ecology. I will discuss. Also, we developed a biomarker database, disease biomarkers for both metabolite and protein biomarkers. The list is here. For computational analysis of diseases, you can utilize this. And also, we have some information about metabolic pathways in motorcycle and cowboy database. And the many chemical compounds are like twins, or maybe very similar to each other. Such information we have gathered here. And now I want to show you how to access the knapsack database. If you click this button core system, you can get such a window like this. And if you enter the name of a metabolite here, for example, if you enter the name of a metabolite here, and then click also the radio button corresponding to metabolite, and then click the list button, you can have the list of the plants that produce sesame and its chemical structure and some other information about sesame. You can also search the database using by entering the name of plant or name of an organism. In such a case, you need to click here this radio button and put the name of the organism here, and then click the list button. So, for example, if we enter Allium cepa, I think all over the world people consume onion, and then click the list button, you can get all the reported in literature. But onion may have more metabolites, but here we have the list only that are reported in scientific literature. So you can get a list of the
Sara, você nos ouve agora? Vou botar a apresentação. Já está na transmissão? Já aparece? Já estou ouvindo direitinho? Ok. Está ouvindo?
Okay. So, sorry for the disturbance. Let us start again. And actually, in the knapsack database, we are trying to accumulate information for linking human omics with plant omics, and also some information of the therapeutic usage of plants, like here. So human omics means the genomics, proteomics, transcriptomics, and metabolomics, and plant omics are the similar terms. So actually, by considering such molecules, I mean metabolites and the genes that are the chains of nucleotides and the proteins that are chains of amino acids, we can have many different types of molecular entities. And based on any context, we can create networks of interaction between. For example, when we humans consume the plants, the plant omics molecules usually interact with our omics molecules and then try to help us maintain good health or to cure us from some disease. In such a way, we can make many different types of interaction networks among the human omics molecules and plant omics molecules. Also, within any one level, for example, human proteins, they interact with each other to do all the molecular functions in the cell. So we can create many networks. And actually, from the beginning of this millennium, there are a lot of research papers that have utilized network algorithms or graph theoretical algorithms to analyze molecular biological data. Even drug development, as you are the pharmaceuticals people, you can see many papers that have developed drugs based on graph theory by analyzing networks of similar structure or drugs in relations and many other things. So network theory is therefore very helpful in analyzing molecular biological data, even pharmacological data. In our lab, we also have developed a clustering algorithm called DP class and DP class O. So now I will try, though this is a matter of computer science people, but you may find it interesting, all right, and then you can utilize it. This tool is also freely available from our website. So roughly speaking, the graphs are big entities like this consisting of nodes and edges. You can create the graph based on any kind of notion you think is useful for your research. Just try to link the different drugs or drug and gene or drug and protein, drug and disease, many types of relations actually can produce different types of graphs, and then you can analyze those graphs to extract useful information. And the clusters in a graph are high density subgraphs. So these might be the clusters in a big graph. So for example, the high density subgraphs in PPI network are usually considered as protein complexes. So initially we try to find out protein complexes in big PPI networks. As you can see here, this is a PPI network of Ecology, the bacteria. Only there are 3,007 proteins here and 11,000 interactions. But we humans have more than 100,000 proteins and millions of interactions among them. So if you just visualize the graph, just by visualization, we cannot get any useful information. Therefore, some algorithm is needed. To find out the dense. So here we are not going to discuss but we try to find out this algorithm by considering density of the graph and also the periphery of the graph. Because here you can see these two graphs both have density 0.5, but this seems to be a single cluster, while this is divided into two clusters. As you can see here one and here is another cluster. So to tackle the periphery, we introduced a concept called cluster property of a node. The cluster property of node F here in the context of this cluster is only 0.2, and the cluster property of node F here in the context of this cluster is 0.57. So you see this measure, this is higher, so this indicates that this is part of this cluster, and this is not part of this cluster because this is lower. So using this concept, we developed and published the graph clustering algorithm and applied it for finding protein complexes in PPI networks a long time ago in 2006. Now many people utilized our tool, and we have a citation index of 537 for this paper. And actually, in this paper, we presented the algorithm, and we have developed a graphical user interface tool, and also we published it here. All these tools you can also have, you can get from this address. It is freely available. Try to use it. Yeah. So our algorithm DP class was compared with many other algorithms. And as you can see, that the precision for DP class was not so good. So we found that we have some limitation to identify overlapping clusters. Therefore, we tried to modify our algorithm, and we also published the DP class O, the overlapping version of DP class. And DP class O can generate many clusters, some of them overlapping. For example, here in this graph, you can see there is a cluster here sandwiched between two bigger clusters, here and here. So this is HEF, also could be detected by DP class O. And maybe in the cell, these three proteins also work as a unit. So it is necessary to find out such clusters also. But DP class itself is also very efficient. It is the 4 major clusters in this graph could be easily identified by DP class, like as it is shown here. Anyway, these graph clustering algorithm also can be applied for pharmacological research. I think we also applied a little bit of that. I will show you later on. And many other people utilized our algorithms for different types of omics research, like metabolomics research, proteomics research, and transcriptomics research. For example, in this work, we tried to find out growth-specific metabolites in E. coli. And then we utilize DB class for classification of ions, into metabolite derivative group. So this is kind of mass spectrometry data. Yeah, DB class was utilized for here. In this work, DB class was utilized to compare with several other methods to find protein complexes in PPI networks. And here, DB class was utilized to find out metabolic correlation network modules in Arabidopsis. And here, DB class was utilized for finding protein complexes in human PPI network. And here, maybe in Oryza sativa, co-expression gene modules in Oryza sativa is rice. DB class was applied. And also, DB class was applied for finding disease pathways, several diseases like Parkinson's disease. Here and here, osteoarthritis, and here, osteoporosis, and here, cancer pathways and biomarkers. And we also applied DB class for finding out relation between plants and diseases based on Indonesian traditional medicine formula. And in this work, we tried to apply DB class for classifying microorganisms based on the volatile organic compounds they emit. And the clustering results imply that the pathogenicity of microorganisms can be linked to the types of VOCs emitted by them. And volatile organic compounds are small molecules. And I think as pharmacological students, you are familiar with chemistry. And VOC has a very important role in chemical ecology, agriculture. For example, the pheromone trap, pheromone is also a VOC. Or for pollination, the flower emit fragrance, that is also VOC. And in human healthcare, I think, VOC also has a good role because to develop non-invasive test diagnostic techniques from the VOC we emit in our breath or urine or sweat, they can be utilized to assess the health condition. So VOC in such a case is very important thing. We have information of all the volatile compounds in our database, chemical ecology database. If you are interested, you can look into that. This is a paper where we applied VOC to relate the structure-activity relations between chemical compounds and their activities. This paper also got a Best Paper Award in 2014 from the Molecular Informatics Nara. Recently, we also tried to analyze the biomarkers of different diseases. We classified the diseases based on protein biomarkers and also metabolite biomarkers. We found these two trees are very similar in some down index not done index. Based on some index, we found that the diseases have very similar classifications we obtained, though by applying metabolite biomarkers and also protein biomarkers. And also, we developed BiClass O algorithm because in many cases the data is not simple graph but bipartite graph. For example, gene expression data, also the genes and the presence or genes and the different experimental conditions are bipartite type. Or many other cases we can find bipartite networks. So we extended the concept of DP-class to apply to bipartite graphs, and we published this work in IEEE Transaction in 2020, and also DP-class or BiClass O, we have developed a tool and called DP-class SBO, and we published it in Software X. So you can get also this tool from the same source. And the bipartite graphs are this type of graphs where two types of nodes. This is a black node and the green nodes, and the edges are allowed only between green and black, no edge between here or no edge between these nodes. And biclusters are densely connected regions in bipartite graphs. That sometimes helps us to find out important drugs. As we have also tried to do some of that job. I will try to show you. And these are the theory of BiClass O. So we are skipping these. Maybe it's not interesting to. We also compare BiClass O with several other biclustering algorithms, BiMax and XMotif and Spectral and so and so. And we use the optimum parameter for each algorithm, and then we applied. For comparison, we utilize three types of data: species VOC relational data, and also gene expression data of yeast, and also synthetic data. The synthetic data means we prepare this data where we have intentionally embedded some biclusters. Here are the biclusters with non-overlapping equal size, variable size and overlapping, and equal size and overlapping, variable size non-overlapping, and variable size overlapping. And also we added a lot of noise here. These dots are noise. And we can show you that for the species VOC, the BiClass O performed very well to find out similar type of species in single cluster, in individual clusters. And also based on expression data, BiClass O performed very well. Here maybe a little bit comparison. And also here BiClass O performed very well. And also for the synthetic data, we found very good result. Here and here you can see. Okay, we use two different types of metrics, and everywhere BiClass O performed very well. Okay, and then I try to talk about ecology a little bit and very quickly. So the ecology, this work also we presented in this paper in IEEE Transaction. Ecology means we try to analyze here, we try to analyze the species VOC relational data. So this is bicluster. We try to find out the biclusters in species VOC relational data, and we have shown that the species can be classified based on the VOCs emitted by them. So now the volatile organic compounds are different species release VOC under some biotic stress. So and become vapor under environmental temperature and pressure. VOCs contain carbon along with many other compounds. I think you are familiar with these things. They have huge environmental and ecological impact as they are the medium of mutual interactions between species. As I told you already, for pollination, the flowers emit something to attract maybe butterfly and other insects. And also symbiosis in the soil between microbe and root plant, also they communicate using VOCs. And when some herbivorous animal try to eat plants, they emit VOCs that invite carnivorous animals to chase the herbivorous animals. That they try to this way they try to survive, and so on and so on. In ecology, there are lots of such communication between species. So VOC is very important. So we and don't want to discuss that much about this. We collected data of So in our data, we have bacteria, fungus, and also insects, and such type of data. And just we created bipartite network between species and VOC. So this species produce this type of VOC, and we, of course, this is a summary of the data. Actually, we can see many species that are linked to only one VOC, because our data is not complete, of course. We just collect from published literature. So we initially deduct some of the species from our analysis. We kept those species that are at least connected to 3 VOC. And as you can see, for some species, we see a lot of VOCs are connected to them. So they are maybe yeast or some other well-studied species. Anyway, we also tried to present a theorem in our paper. This is concerning Tanimoto coefficient, so it's not maybe interesting to you. As you can see here, before clustering, the data is very random. All dots are species versus VOC relation. But after clustering, we could systemize. We could find some big clusters and also many small clusters in the data. And we identified some example clusters here. This is this family, and mostly all are salvia, genus salvia. All those plants have these type of volatile organic compounds. This is a bacteria of this family, and you can say they are producing these VOCs, okay, as a group. And here another cluster showing the plants, and this is the fungi cluster. The fungi also producing some VOCs that are unique to them. And then each cluster, how rich they are with similar family or similar genus species. We tried to find out a p-value for each cluster using the Fisher's exact test. And as you can see from this, there are many clusters with very significant p-values. And I think out of 57 clusters we generated, p-values for 54 are less than 0.05. So this is a threshold usually statisticians consider as significant. And then also we try to analyze the pathways. Usually the similar structure VOCs are produced by similar or pathways. So we try to classify the VOCs according to their structures. Again, we took help from DP class O. We clustered the VOCs according to their structure, and we found, I think, more than 251 type of clusters. This is, for example, cluster 1, long chain hydrocarbon. These are also VOCs, and this is an alcohol type of VOCs, and this is a ring compound, and another cluster is a phenolic compound. So we got more than 200 such clusters. So that means in terms of structure, there are 200 types of VOCs available. And then we try to link that which species are linked to what type of VOCs. In this class here, I think some are bacteria, and these are bacteria, and these are plants, and these are fungus. And as you can see, this is scattered. So not all species use the same type of VOCs. Different species use different type of VOCs. Here, the bacteria, most of the bacteria use less number of VOCs, less types of VOCs, but some bacteria use many types of VOCs. The fungus and then plants, actually, individual plants use many different types of VOCs because they need to survive in adverse situations. And the bacteria also, but as a whole, the bacteria use many different types of VOCs, but any individual type of bacteria use very less number of VOCs. We applied some t-test here and tried to find out the relation between plant and fungus and bacteria and so on and so on. So we got some conclusions, like though individual bacterial groups are associated to a smaller number of VOCs, SSBG is structurally similar VOC group. The total number of VOCs related to bacteria clusters are the highest. And bacteria live under diversified environments from the polar region to the hot spring to different types of vessels, requiring diversified types of VOCs. And plants are associated to more diversified types of VOCs as an individual plant compared to fungi, because plants are more advanced species. A number of pathways converge between plant and bacteria. From here you can see that bacteria are more compared to those between bacteria and fungus and plant and fungus. So maybe evolutionary theory somehow we can try to explain from these findings. And now the tool, you can, if you want to use the graph clustering tools, it's a very simple type of data you can enter. I think if you go here, you can download our tools, and we also have tutorials and I think some manual how to use this, and you can make good figures like this. And by clicking on this here, each rectangle is a cluster, and by clicking on this, you can find out individual clusters. You can display, visualize individual clusters in different window. Okay, and also, for example, here are many clusters. Then you can set some threshold of, for example, in terms of size. Maybe people are interested only on bigger clusters. And also you can filter out based on overlapping thresholds. We have such type of facilities in our tool. And also you can do analysis for bipartite data. So bipartite data, you just give 2-column data. Okay, this entity is connected to this entity, and so on and so on. So this is a tool we developed in our lab. So please try to utilize it. We will be happy. And in case you face any problem, send mail to me. I will try to help you. And we also applied a DP class algorithm for finding out, for example, disease genes and pathways for inflammatory bowel disease. So we did this work in 2017 and 18. I think this was published in 17 and 18. And at the time, I met Professor Hayasi in Thailand, and she asked me to whether we can find pathways of schizophrenia and bipolar disorder using computational research. We said that we can try, and also we tried, and we had a publication. I will show next. So here about this work, how to find disease pathways or disease genes in this is a method. Of course, this is a flow of the method. And what we did here, we collected gene expression data. And then we determined the differentially expressed genes, and also we collected the very well-known IBD-related genes, and then we tried to make a disease-related and PPI network with the HIPPI, by collecting data from the HIPPI database involving these proteins or genes. And then we applied clustering by our algorithm, and we found many clusters, and then which are significant in the context of IBD. So by accumulating IBD genes from different sources, so we try to find out significant clusters, and those significant clusters we utilize to find out new IBD genes and also the pathways associated to IBD. So IBD is actually, there are two types, Crohn's disease and ulcerative colitis. And so from the differentially expressed genes, we found that many of those genes are common, which also implies that these two are very similar diseases. Okay, so then maybe we make the network for degree distribution follow the power law. Most of the biological network usually follow power law degree distribution. And we also did some rock analysis to find out good set of clusters. And also this way, with high frequency pathways, we selected as the pathways for IBD. And these are the 20 genes, new genes. We said that they are IBD related genes here, and 10 pathways that we detected by our methods as related to IBD. And in our paper, we have given details, evidence in support of this finding. And also with Professor Hayasi, I did this work. We tried to find the pathways of schizophrenia and bipolar disorder, starting from the metabolic biomarkers. She detected in her lab, and gave me the list of the biomarkers. So from the biomarkers, we tried to identify the pathway of the biomarkers, and from the pathway, we identified the related genes, and then from the genes, we go to proteins, and then from the proteins, we did the clustering, and from the clusters, we identified the pathways. I think 3 major types of pathways we detected here, and also we found a lot of evidence in support of this finding. With help from Professor Hayasi, we could give a good explanation in our paper. This one we published in scientific report. Okay, and also this is we detected IBD related microRNAs by applying biclass algorithm. in this paper, we try to find out microRNA versus gene bipartite network. We try to do the clusters, and then as you can see, as the students, you can see this is a very easy flow to follow. We make the network by collecting data from the databases, and then we do the clustering, and then we try to find the significant clusters in the context of this disease, and then we can, using the clusters, we can bring some conclusions. So in some sense, this is very easy flow you can follow for other disease or other type of research also. So here we applied biclass O, and currently I am trying to find the antibiotics from among the natural compounds. So as a students of pharmacology or pharmaceutical science, we have this big database of knapsack, more than 50,000 natural compounds. You can try to find out many types of compounds effective against different diseases from the natural compounds by applying such type of information using bioinformatics. We are also trying to find out antibacterial and analgesic and also anti-inflammatory compounds from knapsack database by linking the knowledge of biology with computer science and also convolutional neural networks, deep learning, and these are very interesting research topics nowadays. So as you know, the antibiotic resistance is a big problem. Many people it says that number of deaths caused by the resistant microorganisms will reach 10 million annually by 2050, and already now many people are dying because no drug is acting against some infections, some bacteria. So because of overuse of antibiotics, the bacteria is becoming resistant against existing antibacterial drugs. So it is very important. It is creating stress in the health sector, causing more deaths and also hospitalization, cost of hospitalization, and also the economy. Working people cannot work properly, the economy will also suffer. And additional cost for treatment, so health insurance company or the government have to pay. So that's why we are thinking to find out some natural antibiotics, because only solution is that you give more antibiotics, different types of antibiotics at the same time, so that it can attack the bacteria using different mechanisms. So if we can select antibiotics from natural compounds, they will produce less, I think, side effect. So we already published a first paper here. Based on we use the Indonesian traditional formula. We try to classify the formula that are related to bacterial disease, and we have selected some plants that are, I think, that are good against fighting against bacteria, and we also found much evidence. For example, this plant can fight Ecology infection, and this can fight. I think this is a bacteria. You can say this plant can fight this infection. We found in these references about this. So these are we determined using only JAMA formula, and we find support in the literature in support of our findings. And from the plant level, as you know, in knapsack database we have relation about plant metabolites. So we converted our data from JAMA formula to plant. This was the basic formula in JAMA medicine. So JAMA formula to metabolite, because we have plant metabolite relations in knapsack database. And we also now trying to find out identify the natural compounds that are effective against bacteria. We found, I think, more than 111 firm compounds. Out of them, roughly 50% we could validate using published literatures. Here are two examples. For example, we found this compound as antibacterial compound, and it can fight against this bacteria. What is it called? Bacteria names are very difficult to pronounce. So here and also this is effective against this. So this is antibacterial research. Now currently I am doing, and I already finished based on JAMA formula. Now I am trying to find out antibacterial compounds based on traditional Chinese medicine. And this is our campus map. Almost this is all about my talk, and this is campus map of Nara Institute of Science and Technology. We usually invite foreign students. Also, you are welcome. We also provide some scholarship for good students. But even Japan is an expensive country, but I think nowadays it is not so expensive compared to Brazil or America or other countries, because Japanese economy is stuck for last 30 years. So the prices are increased, but Japan is still same price as it was in before 30 years. So and for foreign students, we provide a hostel for the duration of the study period. It is only $100 per month. And the tuition fee, if you compare to America or other countries, is also very cheap in our university. So even self-finance, you can go and maybe you can work for a while. The Japan government allow working for the foreign students. Okay, if we fail to manage a scholarship, still it is not so difficult to continue your study in Japan nowadays. Okay, and we have schools, 3 major schools, and also interdisciplinary schools: information science, material science, and biological science. Pharmacological students can go anywhere. Anywhere you can do some research. Just search our website for the different labs. We have many international students. For example, also from South America, these students already completed their study. So we and mostly from Asia, of course, also from some from North America and Europe. And this is from Africa. So most students usually come from Asia, Southeast Asia or Asia, Indonesia, Thailand, China, and such countries. So we also, therefore, because the students from South America are very less, we have come up to this point to invite you to go there. Okay. And of course, we will try our best for financial support. But as I told just now, that even self-financing is not so possible nowadays compared to other countries, especially in Europe and America. The tuition fee is quite high. In our case, only maybe $5,000, around $5,000 per semester. Not so much. I came from Dallas. Yesterday in the Dallas City, the tuition fee is $15,000 per semester. 1/3 of that, in our institute. And Nara Institute is located in Nara, of course. The Nara is very near from the big cities like Osaka and Kyoto, and this is Nara Park during the spring, a lot of cherry blossom and free roaming deers. You can feed them, not risky. And there's a big Buddha in Nara of bronze, made of bronze. This statue, they claim it is the biggest sitting Buddha, metal Buddha in the world. I think I finished. Yeah. Came to the end of my talk. Thank you very much for listening.
There's some questions in the chat.
Yeah.
I don't know if you prefer to read them.
Of course.
This is better if you read the question and listen.
The question is in English or in?
It's in English. So we are asking if we can verify between the methods of each protein in this network.
So we can verify the between this entries of each protein in these networks. Yes, of course, we have centrality measures.
Sorry.
Yeah, I come. Yeah. So I understand the question. This is a centrality measures for the nodes. Of course, our tool is basically for clustering. So using our tool, you cannot determine these. But I think using cytoscape, very easily you can determine these quantities for each node. These are the properties of the node. Okay. Cytoscape, and I think there are other tools available also for finding these centrality measures for nodes. Can we verify the impact of the deletion of a specific protein in the network? For example, if we remove the protein from the network, the network structure is modified. Yes, this is also using our tool. It is not possible. But these are not so difficult things. Also, you can remove a protein and course, the structure will change. That means the degree and the centrality measures will change. But these are, I think, cytoscape is a better option for doing this type of analysis. Okay, thank you very much for the nice question. Any other question?
Yeah, I have a question. Professor Fabio, he wants with animal models. Do you think that we can model or use those in bioinformatics for analyzing animal behavior and trying to
Yes, that is possible, but we have to discuss the context. For example, with you last time we discussed, you said that we have biomarker for schizophrenia. Can we move to the final pathway based on this? So I thought about it, and then we took out the flow of all kind of information. So for Professor Fabio, if he had some data and if he had some imagination that we can find out some information, then I can think of a way that my bioinformatics how to do that. Please contact with me. Yeah, I'm happy to collaborate with you.
Okay, so it means that not necessarily we need to have biomarkers and molecules for making errors. It could be animal behavior or some specific.
Yes, yeah. For biomarkers, easily eating chemical fingerprints, we can make a network and we can find out important things into clustering, and then that can lead to some other information.
Okay, thank you. Any other questions?
Professor, nice to meet you. My name is Thais. I was wondering more about the data these hours. For example, if you have a group that are studying a complex protein, and they provide new information about the pathway. This new information can be put on the system, and the system can be updated anytime.
Yes, can you repeat your question? No problem.
Okay, so once you have done the analysis, like a function site reports, can you add new information to there, or you have just to start with the analysis and then first? So you have a new incoming. So if we have some new data, like for example.
In abstract database or?
Yeah, for making those networks.
Networks.
Yeah.
Well, then our one is not the database of protein interaction network. So PPI network, there are many other databases like string and HIP, which we use. So they are adding new information here. And actually now there is millions of interactions, so very difficult to extract good information from here. But you can, they have some I think quality of some measure to assess that or to recognize the quality of the interaction based on that. So you should of course use good interactions for your research. That's important. Otherwise there will be a lot of noise and you cannot find many good things. And our one is only the algorithm that can cluster. That can find the clusters from a network, and the network you extract from a PPI network or the network of molecules or network of even social network, any type of network. You prepare the network, and we just need very simple type of input data, just a edge list. Edge list means protein A interact with protein B and protein C interact with protein B. This type of you can have 10,000 lines, no problem. You enter, and the algorithm will give you which are the group of proteins that are densely connected to each other, or a group of genes, or a group of molecules, or a group of people, a group of business, a group of commodity, anything. The network you need to create by yourself. Okay, and then you enter the network, and then it will give you the cluster. After you get the cluster, you try to see whether this cluster is very interesting or not. Okay. There are many other tools to do that. Or
Any questions? No, I just would like to thank you very much for this presentation.
Thank you very much. So can I take a photo here?
Yes, sure. I have just
Please come here.
so thank you very much. Thank you also for the audience in YouTube.
Yeah, many thanks to the online and offline audiences. for listening to my talk.